In the autumn of 1907, William James stood at the front of a lecture hall at Harvard University, and the afternoon light came through the tall windows at a low angle and lay in pale strips across the wooden floor. He was 65 years old and had spent most of his life thinking carefully about what the mind was and what it could do. The audience before him was composed of people who had come to hear something useful, something that might apply to their own lives when they left the building and returned to the ordinary difficulty of being human. James did not disappoint them. He spoke in his measured and unhurried way about energy, not electrical energy, not the energy of steam engines, but the subtler and less quantifiable energy of human beings going about their days. Most people, he suggested, move through their lives drawing on only a fraction of the mental and physical resources available to them. They stop well before exhaustion. They settle into habits that spare them the trouble of effort. The full range of what a person could summon, James said, rarely gets summoned. He did not offer a number. He did not produce a percentage or a fraction or a figure of any kind. The observation was philosophical, carefully hedged, addressed to an audience he expected to think about it rather than to retail it unchanged. The thin afternoon light held the room. The words left it. What happened to those words over the following decades is one of the more instructive small histories of how ideas travel and what they lose in the traveling. James's lecture was published in an essay called The Energies of Men, and it circulated among readers who found its central claim genuinely useful, not because it told them something scientific, but because it named something they recognized. The feeling that more was available, that the ordinary level of effort represented something less than the possible maximum, was not a claim that required verification. It felt true, and things that feel true have a particular kind of durability in a world that moves faster than any single person's ability to check sources. The essay passed from reader to reader. The qualifications softened with each passing, the philosophical carefulness worn away like the edges of a stone that many hands have handled. By the time it reached the general public in usable form, the hedge had gone, the nuance had gone, and what remained was a premise that people used only a part of what their brains could offer. The premise, still warm and still persuasive, was ready to be measured. Someone, at some point, attached a number to it. No one knows exactly when or exactly who. The number was 10%. Albert Einstein is perhaps the most famous name attached to the 10% claim, which is precisely the kind of attribution that a claim acquires when it needs authority it does not have. The researchers at the Quote Investigator Project, who spend their working hours tracing statements back to their actual sources, have searched Einstein's full archive, his writings, his published interviews, the transcripts of his recorded speeches, the letters held in university collections, and found no authentic version of the statement. Not a paraphrase, not an approximation, not a qualified version of the idea that could plausibly have been simplified into the familiar form. The attribution appears to have been applied after the fact, the way a reputable address is sometimes listed on a product to lend it the credibility the product itself cannot earn. Einstein said many things that were genuinely interesting and genuinely his, and none of them included a claim about 10% of the brain. The process by which a famous person's name becomes attached to a statement they never made is not usually a process of deliberate deception. It moves more slowly than that, and more innocently. Someone paraphrases a real Einstein observation, slightly imprecisely, and the paraphrase circulates. Someone else encountering a claim about untapped mental potential thinks it sounds like something Einstein might have said. It has that quality of plain wisdom, that his actual public statements sometimes carried, and attributes it to him in passing. A third person, finding it already attributed, accepts the attribution without checking, because why would Einstein's name be attached to something he did not say? By the time anyone thinks to ask for documentation, the claim is already established, the name is already welded to it, and the work of disentangling the two requires a particular kind of patience that most contexts do not reward. What the name did for the claim was considerable. It moved a piece of folk wisdom from the register of motivational assertion into something that felt closer to scientific fact. Einstein had understood the atom, or something like it. If Einstein said the brain was underused, then the brain was underused in a way that carried at least the flavor of physics. The claim found its most consequential early home not in a scientific paper, but in a book, and not in the body of the book, but in its preface. Dale Carnegie published How to Win Friends and Influence People in 1936, and the book's preface was written by a journalist and broadcaster named Lowell Thomas, who invoked William James's idea of untapped mental reserves in terms that presented it not as a philosophical hypothesis, but as an established fact about human limitation. Thomas was a skilled communicator who understood that a preface needed to do a specific kind of work. It needed to make the reader feel, before the first chapter even began, that they were about to receive something they genuinely needed. The deficit narrative, the suggestion that the reader was currently operating below capacity, that something important was being left unused, was ideally suited to this purpose. It created a problem and implied a solution in the same sentence. The book that followed would help the reader close the gap between what they were and what they could be. The preface made the reader receptive before they had read a word of the actual argument. Carnegie's book sold in the millions. It sat on bedside tables and office desks and library shelves in countries across the world. A worn copy on a nightstand somewhere in 1937, its preface already creased from rereading, the glass of water beside it, the lamp switched off. That book carried the untapped reserves idea to more people than William James's collected essays had reached in 30 years of circulation, and it carried the idea in the form most useful to the people who would later find other uses for it, as a statement of human deficit, as a gap waiting to be closed, as a problem that implied a market for solutions. The market did not take long to recognize the opportunity. By the middle of the 20th century, a figure of roughly 10% had become a standard piece of furniture in the American motivational landscape. It appeared in speeches, in advertisements, in the promotional materials for correspondence courses that promised to develop the listener's latent mental capacities. The figure had by then acquired a comfortable feeling of factual solidity that owed nothing to any actual research and everything to repetition. When something is said often enough by confident enough speakers in contexts that carry the appearance of authority, it begins to feel verified. The listener does not actively decide to believe it. The belief arrives before the decision is available. At the front of a mid-century sales conference, a speaker gestures broadly at the audience. Men in good suits, seated in rows, their attention given freely, because this is the kind of thing they have driven some distance to hear. Behind the speaker, on a chalkboard, the figure 10% is written in large and looping numbers. There is nothing beneath the number, no citation, no reference, no qualification. The number simply stands there, as though it has always stood there, as though it were the kind of thing that does not require a source, because its truth is obvious to anyone who thinks about it for a moment. The speaker moves on, and no one in the room raises a hand to ask where the figure came from, because the figure already feels like something they have always known. This is the particular power of a well-placed number. A vague assertion, people do not use everything available to them, leaves room for argument, for degrees of agreement, for the natural hedging of conversation. A specific percentage forecloses that room. It implies measurement. It implies that someone somewhere performed the work of quantification and that the number is the result of that work. The listener inherits the implied authority without needing to examine its origin. 10% is specific enough to feel true and round enough to remember. The neurological research of the early 20th century offered what seemed, to non-specialist readers, like a suggestive piece of corroboration. Carl Lashley was a neurologist who spent years conducting a particular kind of experiment. He removed portions of rats' cortices, lesioning specific areas of the brain, and then tested the rats to see which learned behaviors survived. The results were interesting, and genuinely so, for reasons that had nothing to do with what popular writers would later make of them. Lashley found that certain learned behaviors could persist even when a portion of the cortex had been removed. The rat would complete a maze it had been taught, even with tissue missing. This finding pointed towards something real about how memories are distributed across neural networks, about the brain's capacity for redundancy and reorganization, about the way function is not always lodged in a single precise location. What it did not point toward in any responsible reading was the conclusion that large sections of the brain were functionally silent or unused. Lashley was documenting reorganization and distribution, not vacancy. But the leap was not a long one for a reader who had already encountered the 10% figure and was looking, consciously or not, for evidence that would support it. A rat completing a maze despite a missing piece of cortex could be read, if read quickly and without precision, as confirmation that significant portions of the brain were dispensable, that they were there but not essential, present but not needed. This is not what Lashley's findings established, and it is not what Lashley argued. The popular interpretation moved ahead of the data, as popular interpretations often do, and settled into a form of the claim that felt scientifically endorsed without being so. In a laboratory at a university, a small rat navigates a simple maze, left, then left again, then right, then the food reward at the end of the run. It has done this many times. A section of its cortex has been removed in an earlier procedure, and the rat completed the maze correctly regardless. For Lashley, this was a finding about distribution, about the way learning is stored, not in a single location, but across a broader network of tissue. For the popular writers who encountered the result at some remove, the rat in the maze became evidence of something larger and less careful, that substantial portions of the brain could be absent without consequence, which seemed, to that reading, to confirm that substantial portions of the brain must ordinarily be doing nothing much at all. The two readings are not equally faithful to the data, but in the middle decades of the 20th century, faithfulness to data was not the quality most in demand from the 10% claim. What was in demand was utility, and the claim was extraordinarily useful. Memory courses, in particular, found the claim congenial. The premise that the average person was using only a tenth of their available cognitive capacity was exactly the kind of statement that made a memory course seem not merely helpful, but almost obligatory. The gap between current performance and theoretical potential was wide enough, the claim suggested, that any person who genuinely wished to improve had enormous room to do so, and the course being advertised was one way to begin closing that gap. The course did not need to be demonstrably effective in any specific scientific sense. It only needed to be offered in the context of a deficit that felt real, and the 10% figure made the deficit feel as real as anything in the listener's experience. A print advertisement from the 1950s shows a diagram of the human brain, rendered in clean graphic lines in the style of that era's scientific illustration. The diagram is shaded. Most of the brain, 80, perhaps 90%, is filled with a flat gray tone and labeled in neat type, dormant. A smaller portion at the front, or the side, or wherever the designer thought it most visually effective, is left white or shaded differently and labeled with something along the lines of active or in use. Beneath the diagram, the advertisement promises that a correspondence course, available by mail and reasonably priced, will help the reader develop the unused portion. The logic is entirely visual. The brain is mostly gray. The gray portion is dormant. This is presented as though it were anatomical fact. Below the brain diagram, the ordering information, there is a particular quality to that kind of advertisement that rewards a moment of attention. It does not fabricate the brain diagram in a spirit of obvious dishonesty. The designer who drew it was probably working from a genuine belief that the information was correct, because the information had been circulating for long enough, in enough authoritative-seeming contexts, to feel correct to a careful designer. The claim had passed through enough hands that it no longer carried the stigma of the unverified. It had become, in the cultural sense, simply known, the kind of thing that everyone already knows, the kind of thing that does not need sourcing because its source is assumed to be common knowledge. This is perhaps the most interesting thing about the 10% myth as a historical object, the manner of its settling. Most false beliefs circulate because someone is promoting them, because there is an interest being served by their acceptance. And there were certainly interest being served here: the memory courses, the self-improvement programs, the motivational speakers, whose most compelling opening move was to make their audiences feel that something important was being wasted. But the settling of the claim, the way it stopped feeling like a claim and started feeling like a background fact, was not primarily a story of cynical manipulation. It was a story of useful information finding willing hosts. William James, at the front of that Harvard lecture hall in 1907, was not wrong that people habitually operate below their potential. He was describing something recognizable about human psychology, about the gap between effort and its possible maximum, about the ease with which habit substitutes for exertion. His observation was careful and qualified, and addressed to an audience equipped to hold it in that form. The forms in which it was inherited by later readers and later audiences were progressively less careful, progressively less qualified, progressively further from the specific intellectual tradition in which the observation had been made. Dale Carnegie did not set out to propagate a myth. He was writing a practical book about human relations, and the preface by Lowell Thomas, invoking James's idea of untapped potential, served the book's purpose without anyone in that editorial transaction stopping to ask whether the claim could be verified. The motivational speakers did not consider themselves to be deceiving their audiences. They were drawing on what seemed to them a credible premise, sourced to credible names, in service of a message they believed to be genuinely helpful. Even the advertisements, with their shaded brain diagrams, were produced by people who had absorbed the claim from the general cultural air without examining it. The figure 10% moved through the 20th century the way a confident rumor moves through a community. Quickly, usefully, and with a self-sustaining social logic that made verification feel unnecessary. To check the figure was to imply that the person who had stated it might be wrong, and the person who had stated it was usually someone whose authority in the room was not easily questioned. The famous author, the motivational speaker, the advertisement endorsed by the scientific-looking diagram. Checking requires a particular kind of friction, a willingness to interrupt the smooth transaction of shared belief for the uncomfortable work of verification. Most people, in most moments, choose not to generate that friction. And so the number persisted. It persisted in part because it was the right kind of number, large enough to imply an enormous and exciting gap between present and possible, small enough to feel specific and measured. 90% dormant is a finding dramatic enough to want to believe, and precise enough to cite in conversation. If the claim had been that humans used some fraction of their brains, or that a great deal of potential goes untapped, it would have had much less traction. Fractions are vague, percentages are authoritative. The number 10 did the work that no amount of careful philosophical qualification could do, because it arrived with the implied authority of measurement without requiring any of the actual labor of measuring. By the time the claim reached the second half of the 20th century, it had been so thoroughly absorbed into the popular vocabulary of self-improvement that its origins were essentially invisible. It was not a claim you needed to source, because it was simply a known thing, the kind of fact that everyone had heard and no one had seriously questioned. The academic literature had not endorsed it, but the academic literature was not, for most of the claim's audience, a primary reference. The endorsement it had received from the cultural circulation of James's idea, from Carnegie's enormous readership, from the repeated confident assertion of speakers and advertisers, was sufficient for the purposes to which the claim was being put. A claim about untapped potential does not need to be neuroscientifically accurate to be motivationally effective. This is perhaps the most important thing to understand about the 10% myth's long life. It survived not because it was true, but because it was useful. And it was useful in precisely the way that a deficit narrative is always useful. It created a gap, named a lack, and implied that the gap could be closed by effort, by training, by the right course of study, or the right motivational framework. The people who heard the claim and acted on it were not wrong to act on it. The underlying message that human beings are capable of more than they ordinarily manage, that habit and comfort limit effort more than capacity does, was not a falsehood. It was James's original observation, stripped of its careful form. The number was false. The intuition behind it was something older and less easily dismissed. In lecture halls and conference rooms, in printed advertisements, and on the pages of best-selling books, the 10% figure accumulated the weight of a fact without ever having been established as one. It settled into the general cultural knowledge the way a piece of furniture settles into a room, gradually, without announcement, and in a way that eventually makes it difficult to imagine the room having ever looked different. No one put it there in a single decisive moment. It arrived through a series of small inheritances, each step small enough to seem unremarkable, the cumulative distance from origin to settled fact, visible only in retrospect. Carl Lashley's rats ran their mazes. Albert Einstein's name lent its borrowed weight. Dale Carnegie's readers turned the pages of their preface in lamplight, ready to believe that something important in them had not yet been used. And the motivational speaker at the conference, having written his number on the chalkboard in the confident hand of a person stating a known thing, had by now turned back to face his audience, the large looping 10% standing behind him in plain chalk. There was nothing beneath it. No citation, no researcher's name, no date, no paper in any journal that could be tracked down and examined. The number stood on its own, which numbers rarely do when they are genuinely earned, and it stood there with the particular authority of a thing that no one in the room was going to question. It would remain in that condition, solid seeming, useful, unexamined, circulating freely for many more decades. The chalk would be erased from that particular chalkboard, but the number would go on being written elsewhere, in other rooms, for other audiences, by speakers who had inherited it from speakers who had inherited it from a slow accumulation of cultural transmission that stretched back, in ways none of them knew, to a philosopher standing in autumn light, speaking carefully about energy, offering a thought he never expected to be simplified into a figure, addressed to an audience he never imagined would one day include the entirety of the motivational industry. The thought left the room in Cambridge and traveled a long way. It collected a number somewhere along the route, and the number collected a name, and the name collected the quiet, unearned authority of a fact that had been stated so often in so many rooms that the question of where it came from had simply stopped being asked. In a hospital research wing, sometime after 9 in the evening, a functional MRI scanner fills its small room with a sound that is less a noise than a physical fact, a rhythmic knocking, low and regular, like a freight train passing at a distance over old tracks. The sound comes from the magnetic gradient switching on and off in rapid sequence inside the machine's bore, and it has been going on long enough that the technician in the adjacent control room no longer hears it as a sound at all. She watches a monitor instead. A volunteer is lying inside the scanner, perfectly still, a small screen suspended above her face, displaying a single sentence of ordinary prose. She is reading it. That is all she has been asked to do. And on the monitor in the control room, color is spreading across the image of her brain, the way light spreads across a surface when a lamp is slowly turned up, not all at once, not explosively, but steadily, in clusters, in regions that have names and addresses and documented responsibilities, each one brightening as the tissue beneath it draws more oxygen from the blood flowing through it. The signal the scanner is detecting is called the bold response. The full name is the blood oxygen level dependent signal, and it rises in regions where neurons are consuming oxygen at a higher rate than usual, which happens when those neurons are more active than usual, which happens when the brain is doing something that requires them. The technique for detecting this signal is called functional magnetic resonance imaging, and it does not measure electrical activity directly. What it measures is the ratio of oxygenated to deoxygenated hemoglobin in the small blood vessels supplying each region, because oxygenated and deoxygenated hemoglobin have different magnetic properties, and the scanner can tell them apart. The neuron fires. The local blood vessels dilate slightly to bring it more fuel. The ratio shifts. The scanner sees the shift. The image changes color. The technician watches. The volunteer reads her sentence in the dark. What the monitor shows in those first minutes is not a brain with most of its territory quiet and a small lit corner doing the work. The image is not dim with a single bright point. It is broadly active in the way a city seen from above at dusk is broadly active, some districts brighter than others, some traffic heavier on certain roads, but nowhere fully dark, nowhere switched off, nowhere waiting to be needed for some special occasion that has not yet arrived. The reading volunteer's visual cortex is engaged, processing the shapes of letters. Her language areas are engaged, parsing the sentence into meaning. Her working memory is holding the beginning of the sentence in mind, while her eyes reach the end of it. Her attention network is maintaining the task. And beneath all of that deliberate, legible activity, a quieter metabolic hum is running, the baseline cost of keeping every region organized and ready, which is itself a considerable cost, and does not stop when the task does. The neurosynth database represents something that took decades to assemble. It is an aggregated repository drawing on more than 15,000 individual neuroimaging studies, each one documenting which brain regions showed significant activation during which specific tasks or conditions. The database was built to make it possible to ask questions across the entire literature at once, rather than one study at a time. And the kind of question it is particularly well suited to answer is whether any given brain region has ever, across 15,000 experiments and many thousands of volunteers, been found to simply sit out, to show no activation under any condition, to remain across the full range of human mental life that the studies collectively cover, perpetually and completely silent. No major brain region has been found to do this. Not one. The database has entries for language and memory, and emotion and movement and perception and spatial reasoning and social judgment and mathematical processing and dozens of other categories. And the finding that runs through all of them is that every anatomically distinct area the research has examined shows activation under the right circumstances. The right circumstances differ by region, but the circumstances exist. This is worth sitting with for a moment, not because it is a dramatic finding in the way that word usually implies, but because it is a thoroughly documented one. 15,000 studies is not a single experiment whose results might be fluke or artifact. It is a literature. It is a body of work built over decades in laboratories on several continents by researchers who were not, in most cases, trying to disprove any particular popular belief. They were studying memory, or attention, or fear, or language, and the cumulative picture that their individual findings, assembled when placed together, was simply this: the brain is in use. All of it. Not all of it at once, in the way that not every road in a city carries its maximum traffic at every moment, but all of it across the range of circumstances that make up a human life. A positron emission tomography scanner works by a different principle. The volunteer swallows or is injected with a small quantity of a radioactive glucose analog, a molecule that behaves chemically like glucose, which the brain uses as its primary fuel, but that carries a radioactive tag that the scanner can detect. The analog is taken up by brain tissue in proportion to how metabolically active that tissue is. Regions that are working harder consume more of it. The radioactive decay of the tag produces photons that the scanner catches and uses to construct a three-dimensional image of glucose consumption across the brain. The brighter the region in the resulting image, the more fuel it is burning. And what PT scanning has shown across many studies of subjects in many different states is that the brain maintains roughly 80% of its maximum metabolic rate, even during deliberate rest, during the condition that most closely resembles doing nothing. The brain, asked to sit quietly and let the mind wander, continues consuming fuel at a rate that would be considered high activity in almost any other organ. It does not take a break when the task stops. It takes a different kind of task. A pet image of a resting brain has a quality that is easy to underestimate if the only frame of reference is the idea of dormancy. The image is not dim with a few lit patches. It is warm throughout, in the way that a room is warm when the heat has been running all day, not blazing in every corner, but nowhere cold, nowhere abandoned by the system that maintains it. The surface of the brain in the image is suffused with the quiet metabolic work of tissue that is neither solving a problem in the conventional sense nor sleeping, but is simply running, maintaining the electrochemical gradients that let neurons fire when they need to. synthesizing neurotransmitters at the synapses, keeping the ion pumps working, sustaining the infrastructure that makes every other kind of thinking possible. This is not spare capacity sitting idle. This is the cost of being organized and ready, which is a real cost borne continuously, and it is being borne across the whole organ at once. The sleeping brain presented researchers with something that required some adjustment to how they had been thinking about the brain at rest. When neuroimaging technology became capable enough to study subjects who were actually asleep rather than simply lying still with their eyes closed, the images that came back were not images of a quieting organ winding down for the night. The amygdala, a small dense structure tucked into the medial temporal lobe, involved in the processing of emotionally significant stimuli, showed activation during REM sleep at levels comparable to, and in some studies exceeding, those observed during alert waking. The anterior cingulate cortex, which participates in emotional regulation and the monitoring of internal states, was similarly active. The visual cortex was generating its own images without any input from the eyes. The brain was not resting in the sense of reducing its operations. It was conducting different operations with different regions more prominent than during waking, and the overall pattern of activity was not a diminished version of the daytime brain, but a reorganized one, busy with work that waking does not leave room for. The question of what that work consists of has been one of the more productive areas of sleep research in the last 3 decades, and the answers have tended to converge on the same basic picture. The sleeping brain consolidates what the waking brain learned. It processes emotional material that was too charged or too complicated to integrate cleanly during the day. It replays motor sequences to strengthen them. It clears the metabolic debris that accumulated during waking hours. These are not minor housekeeping tasks. They are the activities that make the next day's waking cognition possible, and they require broad, organized neural engagement to accomplish. The brain asleep is not the brain switched off. The brain asleep is the brain working on a different shift, with a different set of priorities, in the same building. Clinical neurology arrived at a similar conclusion through a different kind of evidence, not by watching the brain in action through scanners, but by observing what happens when specific parts of it stop working. The clinical record is vast. It has been accumulating since the 19th century, when physicians first began making careful, systematic observations of the relationship between the location of a lesion and the specific nature of the deficit that followed. And what the record shows, mapped across generations of patients and hundreds of distinct brain regions, is a picture in which virtually every major area, when damaged, produces a predictable and specific loss. Not a general decline, not an overall dimming of function. A specific, nameable, reproducible deficit tied to the location of the damage. Damage to a region in the left posterior frontal lobe, an area that came to be called Broca's area after the French physician who documented its role in the 1860s, produces a characteristic disruption of speech production. The patient understands language, they can read, their comprehension is largely preserved. What they lose is fluent, organized speech, the ability to produce grammatically organized sentences with something like ease. The words are in there and the meanings are in there, but the machinery for assembling and delivering them has been compromised. The deficit is precise, the location of the damage is precise. The relationship between the two is reproducible across patients with similar lesions. It does not look like the behavior of a region that was doing nothing before it was damaged. A different region, positioned further back and higher up in the left hemisphere, in the posterior superior temporal gyrus, was identified by the German physician Karl Wernicke in the 1870s. Damage here produces a different kind of language disruption. Speech is fluent, grammatically organized on the surface, but the words are the wrong words. Comprehension is severely impaired. The patient speaks easily and at length, but the content is difficult to follow, full of substitutions and invented words and phrases that sound like language without quite being it. Two regions, two different deficits, two different precise relationships between a small piece of tissue and a specific function. The specificity itself is the argument. If those regions had been doing nothing, damaging them would have changed nothing. The cerebellum sits at the back and base of the brain, beneath the large hemispheres of the cortex, and for a long time it was understood primarily as a motor structure, the part of the brain responsible for the smooth coordination of movement, for the timing and balance that let a person reach for a glass without knocking it over or walk across a room without falling. Damage to the cerebellum produces a characteristic set of movement difficulties, a gait that is wide-based and unsteady, difficulty with fine coordinated movements, a tremor that appears when trying to reach for something rather than at rest. The deficit is called cerebellar ataxia, and it has been documented in patients with cerebellar damage from many causes, and the consistency of the picture across patients has left little doubt that the cerebellum is doing something necessary for movement. What has become clearer more recently is that the cerebellum is doing other things as well. The cerebellum contains, packed into its relatively small volume, more than half of the brain's total number of neurons, a density that is itself suggestive of how much processing is happening there. And neuroimaging studies have found that the cerebellum is active during tasks that have nothing to do with movement in any obvious sense, during working memory tasks, during language processing, during sustained attention. The connectivity between the cerebellum and the prefrontal cortex, which handles planning and judgment and impulse control, has been documented in both structural and functional imaging studies. The cerebellar contribution to cognition beyond movement is still being characterized, and the picture is continuing to develop, but the direction is consistent. The structure has more responsibilities than its traditional description captured. A resting brain, if such a thing were possible to straightforwardly define, is not a brain that has gone quiet. This was established clearly enough through imaging studies in the early 2000s that it changed how researchers thought about what the baseline condition of the brain actually meant. Marcus Reichel and colleagues, working to understand why certain brain regions showed decreased activity when a subject began a deliberate cognitive task, found that those same regions showed robust, consistent activation during apparent rest. The regions were not simply failing to respond to the task, they were doing something else, something that the task interrupted. The network they identified came to be called the default mode network, and its activity during rest has since been associated with autobiographical memory, future planning, social reasoning, and the kind of undirected mental activity that in everyday life goes by the name of mind wandering. The default mode network activates when a person lies in a scanner between tasks, letting the mind go where it goes. The medial prefrontal cortex brightens, the posterior cingulate brightens, the angular gyrus, tucked at the junction of several lobes, brightens. The network as a whole produces a pattern of coordinated activity that is not random noise, but organized, reproducible, and functionally significant enough that disruption of its normal patterns has been associated with depression, Alzheimer's disease, and schizophrenia, among other conditions. A network whose disruption has consequences that specific is not a network that can be dismissed as filler activity or background noise. It is a network with a job. The job involves the interior work of being a person in time, remembering what has happened, imagining what might happen, modeling what other people are thinking and feeling, and it runs most fully when nothing else is demanding attention. The lesion map in a neurology textbook is a particular kind of document. It is not a diagram of what researchers believe the brain does in theory. It is a map drawn from observations of actual patients over more than a century, each entry the result of a specific case in which a specific loss of tissue at a specific location produced a specific documented change in function. The map has entries for the frontal lobes and the parietal lobes and the temporal lobes and the occipital lobes. It has entries for the subcortical structures, including the hippocampus and the amygdala and the basal ganglia and the thalamus. It has entries for the cerebellum and the brainstem. The entries describe what was lost when the tissue in each location stopped working correctly, and the description is always particular. This kind of memory, this aspect of movement, this element of emotional processing, this component of perception, the spatial reasoning that lets a person navigate through a familiar building and know without consciously thinking about it where the stairs are and which way to turn. Damage to the parietal cortex, particularly on the right side, can disrupt this in ways that are striking and consistent. A patient with a lesion in the right parietal region may lose awareness of the left side of space entirely, not because they cannot see it, but because the brain's capacity to attend to it and represent it has been compromised. The condition is called hemispatial neglect, and patients with it may eat only from the right side of their plate, draw only the right side of a clock face when asked to draw a complete one, and fail to register people or objects in the left half of their visual field. The specificity of the deficit, its relationship to a specific location, a specific function, a specific failure to represent a specific region of space, makes the argument about redundancy difficult to sustain. The thalamus sits near the center of the brain, a small paired structure about the size of 2 walnuts, and it acts as a relay station for sensory information on its way to the cortex. Almost all sensory signals except olfaction pass through the thalamus before reaching their cortical destinations. Damage to the thalamus, depending on its precise location, can disrupt consciousness, sensation, sleep, attention, and memory, sometimes in combinations that are difficult to fully characterize because so many systems run through the same small territory. The thalamus is not a passive conduit. It participates in gating and modulating the signals it passes, and the nature of that participation has turned out to be more complex and more consequential than earlier models suggested. Like the cerebellum, it does more than its simpler historical descriptions implied. Back in the control room, the technician is watching the monitor, and the volunteer is still reading. The sentence on the screen above her face has been replaced by a new one. The color across the brain image has shifted slightly. One cluster has brightened, another has quieted a little, and a third has come up that was less prominent before. The shift is subtle, and it is happening in real time, and the pattern it produces is nothing like the image that a 10% claim would predict. There is no large gray region waiting to be activated. There is no dormant territory holding its untapped potential for some future occasion. What the monitor shows is a brain distributing its work across an architecture that has been shaped over a very long time for exactly this kind of continuous, differentiated, expensive operation. The cost is worth noting one more time because it matters to the picture. An organ that consumed the body's resources at the rate the brain does across the full span of a human life. would not persist in its current form if the body of evidence from the last century of neuroscience were consistent with 90% of it doing nothing. The metabolic argument and the imaging argument and the clinical argument do not each prove the same thing independently of one another. They converge. They describe the same organ from different angles and arrive at the same picture. The PET scanner showing 80% metabolic activity at rest, the functional MRI showing distributed activation during ordinary tasks, and the neurology textbook documenting specific deficits following damage to specific regions. These are different instruments pointing at the same fact, when the fact they are pointing at is not complicated. It is simply that the brain is in use, and always has been. A neurologist examining a patient who has suffered damage to a small region in the posterior left hemisphere and can no longer recognize familiar faces, the condition called prosopagnosia, is not encountering something that ought to be controversial. The relationship between that specific location and that specific function has been documented across patients with similar lesions. The function existed before the damage. The damage changed it. The location of the damage predicts the nature of the change. And the implication is the same as it is for every entry in the lesion map. The tissue that was damaged was doing something, and what it was doing was particular to it, and its loss changed the person in a way that nothing else in the brain could fully compensate for. That is what the clinical record shows, across a century of observations, across hundreds of different regions and hundreds of different deficits. The map has entries for every part, and what the entries say, consistently and without exception, is that every part was occupied. The volunteer in the scanner has finished reading her sentence. Her task has ended. The technician in the control room watches the monitor as the deliberate language activity settles, the regions that were most prominent during reading becoming somewhat less so, the pattern shifting as the task releases and the brain's resting configuration begins to reassert itself. The visual cortex quiets slightly, the language areas step back a little, or Haspel-Simdock network begins to come up across several regions simultaneously, the medial prefrontal cortex and the posterior cingulate brightening together in the particular coordinated way that marks the transition from task to open attention. The brain has not gone quiet. It has changed what it is doing. It has moved from reading someone else's sentence to whatever private drift of attention the mind settles into when the specific demand of a task is lifted and the interior work begins. The lesion map, a diagram in a neurology textbook, its pages dense with pencil-fine annotations, and the names of regions arranged in careful anatomical order, records something that accumulated slowly, one patient at a time, over more than 100 years. Each entry in the map was arrived at through observation that was careful enough to be reproducible, documented well enough to be compared with other cases, and consistent enough across different patients and different clinicians to harden from anecdote into evidence. The map is not finished. New regions are still being associated with functions that were not fully characterized before, and the understanding of how regions interact and influence one another continues to develop. But the direction the map has been moving since it began is not toward more blank space. It is toward more entries. Every decade of clinical neurology and neuroimaging research has added more detail to the picture of a brain in which every part is doing something, and the detail has not been added in any single corner. It has been added everywhere, across all the regions and all the lobes and all the subcortical structures, filling in a picture that looks less like a landscape, with most of its territory empty, and more like a map that simply keeps getting more specific, more annotated, more crowded with the particular work of particular places. Every region marked, every entry occupied. No blank spaces left where the word unnecessary might honestly be written. In a university anatomy department, on a quiet weekday morning, a researcher lifts a human brain from its container with both gloved hands, settling it carefully in her palms. It is smaller than most people expect. It weighs roughly what a large grapefruit weighs, somewhere between 1.5 and 2 pounds, cool and dense and still, its surface folded into soft ridges that hold their shape without assistance. She has handled many of them. She still pauses briefly each time, what she holds in her hands consumed, across the entire life of its owner, approximately a fifth of every calorie that person ever ate. Every meal, every piece of fruit, every slow cup of tea, 1 portion in 5 went here, to this quiet object, this 2% fraction of a human body that ran on 20% of the fuel. The arithmetic alone is worth sitting with for a moment. The brain's proportion of the body is modest. Its proportion of the body's energy bill is not. Most organs draw from the body's resources in rough correspondence with their size. Larger organs tend to use more, smaller ones less, and the accounting is reasonably tidy. The brain breaks this arrangement. It weighs less than a bag of flour and demands more energy than organs many times its size. The heart, working continuously through every hour of every decade of a life, uses considerably less. The kidneys, the liver, the lungs, all of them essential, all of them expensive to run, and none of them matching the brain's per gram appetite. The organ sitting in the researcher's hands is by the measure of energy consumption among the most costly structures in the human body. What makes this worth noting is not the number itself. The number matters because of what it implies. A body allocating resources is making biological decisions, decisions shaped over millions of years by what worked, by what kept organisms alive long enough to reproduce, by what turned out to be worth maintaining, and what turned out not to be. When 20% of the body's resting energy flows toward a single organ, that organ has made a compelling case for its existence. It has done so continuously. It has done so in every generation. The cost has been paid without interruption for as long as the human lineage has existed, and the brain has not been reduced or restructured in any direction that suggests the body ever considered it a poor investment. On a plate of food that represents a full day's caloric need, the brain's share would fill roughly 1/5 of the surface. Set that portion aside. The rest, the muscles, the skeleton, the digestive system, the heart, the immune response, the repairs and replacements happening constantly in the skin and the gut and the blood, that all lives in the remaining 4/5. The brain's portion sits alone in its corner of the plate, quietly insisting on its share. It does not adjust much based on whether the person is running or sitting. It does not significantly reduce its demand when the body is at rest. It draws on the available energy in good times and in lean ones, maintaining what it needs regardless of what else is happening. The fuel it prefers is glucose. Under ordinary circumstances, the brain will not accept substitutions. Most tissues, when glucose becomes scarce, can turn to fatty acids and other alternatives. The body has mechanisms for this, and many organs make the shift without great difficulty. The brain is less flexible. It operates almost exclusively on glucose under normal conditions, and the supply must be continuous, not intermittent, not approximate. The neurons running in the brain at any given moment require a steady stream of glucose arriving through the bloodstream, and a significant interruption in that supply produces consequences that become apparent very quickly and in a very specific order. The organ is not designed for shortages. It is designed for continuous provision, and it has arranged its relationship with the body accordingly. To understand why the demand is so high, it helps to think about what a single neuron does, just to remain a neuron, just to maintain the basic conditions that allow it to fire at all. A neuron at rest is not actually resting, in the sense that most of the word implies. Its membrane is charged. There is a voltage difference between the inside and outside of the cell, maintained against the constant tendency of ions to drift toward equilibrium. Left alone, that gradient would collapse. The sodium and potassium ions on either side of the membrane would redistribute, the charge would equalize, and the neuron would lose the very condition that makes signaling possible. So the membrane does not leave things alone. Proteins embedded in the cell membrane act as pumps, moving sodium ions out and potassium ions in against their natural gradient, burning ATP molecule by molecule to keep the system charged. This is happening constantly. Not occasionally, not when the neuron fires, constantly, in every neuron, including the ones that appear quiet. Adenosine triphosphate is the molecule the body uses for immediate energy transfer. When a neuron burns it, the neuron gets a small burst of work done, ions moved, signals sent, neurotransmitters built or broken down. When the work is finished, the ATP must be replaced, which requires glucose and oxygen. A single neuron burning ATP to maintain its membrane potential through an ordinary waking hour consumes an amount of energy that sounds small until it is multiplied by the number of neurons doing the same thing simultaneously. The brain contains somewhere in the range of 86 billion neurons, and most of them are not actually quiet, most of the time. The ion pumps are running across all of them, the membranes are charged across all of them, and the cost is accumulating in every one of them at every moment. And every moment, this is before any of them fires in response to anything, before any sensory input arrives, before any thought takes shape, or any memory is reached for. This is just the standing cost of being ready. The mitochondria inside neurons are specialized for exactly this. A synaptic terminal, the small swelling at the end of a neuron where signals pass to the next cell, is packed with mitochondria, dense little structures folded internally to maximize the surface area available for generating ATP. During an active moment, when the synapse is firing and refiring in rapid sequence, the mitochondria in that terminal are running at high output, generating ATP as fast as the cellular machinery allows. The glucose arriving through the local blood supply is being consumed in real time. The oxygen is being consumed in real time. What is visible from outside the skull as a thought, a reflex, a moment of attention, is at the level of the synapse a sustained metabolic event in a space too small to see without magnification. There is a network of activity in the brain that runs during apparent rest. When a person sits quietly, doing nothing in particular, not solving a problem, or remembering anything specific, or planning anything deliberate, a set of brain regions remains robustly engaged, consuming energy, sustaining activity, doing something for which the resting body seems to provide no obvious occasion. This network consumes something close to 80% of the brain's maximum metabolic rate, even in this apparently idle state. The organ does not stand down between tasks. The base cost of running the brain in its ready, organized, background active state is already close to what it would cost during engaged, deliberate work. The increment added by conscious effort is real, but it is added to a base that was already high. This resting activity is not noise. It is not the idle spinning of a system that has nothing to do. The regions engaged in this default state are maintaining something some form of ongoing internal processing that persists precisely because the brain has work to do, even in the absence of a specific task. The cost of keeping the system organized, primed, and continuously processing is the majority of what the brain spends. The deliberate, focused moments that feel like when the brain is really working are metabolically almost a footnote added to the bill that was already due. Think for a moment about what a biological system looks like when it contains genuinely unused capacity. Evolution has a particular way of handling parts that cost resources without returning proportionate benefit. The pressure is slow and patient, and it works across generations rather than within a single life, but over sufficient time it is effective. Organs that lose their usefulness do not simply persist unchanged forever, they tend to shrink. Their cellular machinery becomes less elaborate. The investment the body makes in building and maintaining them decreases, generation by generation, as individuals who dedicate fewer resources to unused structures free up energy for other things. The fossil record and the living world both show many examples of this process working through to its conclusion. In the cave systems of the Yucatan Peninsula, in river passages that have not seen sunlight for hundreds of thousands of years, there lives a species of fish that has been in those caves long enough for the darkness to leave a physical record on its body. The cave fish has no functional eyes. More precisely, it has eye sockets where eyes began to form during embryonic development and then stopped. The process of building ocular tissue starts and is arrested, and the result is a smooth, sealed surface where a functional eye would have been. Maintaining a working eye involves the ongoing production and maintenance of specialized cells, the preservation of delicate optical structures, the development of the retina and the neural pathways that connect it to the visual cortex, and the continuous supply of blood and nutrients to keep those structures alive. When the environment provides no light, and the animal navigates entirely through other senses, the cost of maintaining all of that goes unreturned. Over many generations, individuals with slightly less investment in eye development turned out to be no worse off than individuals with fully developed eyes, and possibly slightly better off, since resources not spent on unused eyes could go elsewhere. The result, visible in the fish today, is an organism whose body has written off the cost of vision because darkness made it unnecessary. This is how the logic of biological investment works, expressed in a living animal. The cave fish did not decide to stop building eyes. No individual fish experienced the change. It happened across generations through the slow accumulation of small differences in how resources were allocated, until the result became visible as a physical feature of the species. The eyes went away because the darkness of the cave made them a cost with no return. And the eyes went away completely, not reduced to 10% function, not maintained at a minimal level just in case, but closed off and sealed and repurposed, because that is what happens to biological structures whose value falls to zero. Against this background, the idea that 90% of the human brain might sit idle becomes difficult to reconcile with how biological investment works. The brain is an expensive structure. Building it during development requires enormous resources. Maintaining it across a lifetime requires a continuous and disproportionate share of the body's energy budget. The skull thickened and hardened around it over the course of primate evolution, investing bone and calcium in protection for this particular organ. Specifically, if 90% of the brain were genuinely dormant, not occasionally quiet, not resting between tasks, but truly and consistently unused, then the evolutionary pressures that sealed the cavefish's eyes would have had many millions of years to work on that 90%. Resources devoted to maintaining dormant tissue would have been available to individuals who maintained less of it. Over sufficient generations, unused brain tissue would have been expected to follow the same path as unused eyes in a lightless cave. The fact that the brain has instead expanded across the primate lineage, growing larger relative to body size at each major step, with the skull tracking that growth protectively, suggests that the opposite pressure has been operating. Looking at the arc of primate brain development, what becomes visible is something like a long, slow biological argument in favor of more. The brains of the earliest primates were small relative to body size. Over the course of primate evolution, spanning tens of millions of years, brain-to-body ratio increased at nearly every major branch of the lineage. The expansion was not uniform across all parts of the brain, but it was consistent in direction. Structures associated with higher cognition grew. The cortex folded inward on itself to accommodate more surface area within the same skull volume, a solution to the physical problem of growing more brain than a fixed skeletal container could easily hold flat. The skull itself grew in response, accommodating the expanding tissue rather than constraining it, adding to the biological investment in protection. All of this happened and continued happening and reversed in essentially no direction across the whole long span of that lineage. Evolution is not a deliberate system. It does not set out to optimize anything. It is simply the accumulated result of differential survival across generations, and over long enough timescales, it tends to reflect what has been consistently worth having. What the primate lineage shows is that more brain has been consistently worth having. Not occasionally, not in some environments, but not others. Consistently, across very different climates and ecological conditions and social arrangements, more brain turned out to be worth the cost. The lineage kept paying it. The protection around it kept being built. The tissue itself kept expanding. A useful way to think about this is in terms of what the body has chosen to protect. The skull is dense, curved, and structurally sophisticated. Its architecture distributes force across a large surface, reduces the risk of fracture at any single point, and encloses the brain in a rigid container that moves as the head moves without flexing or compressing the tissue inside. Building this is not without cost. Calcium and phosphorus and collagen and cellular maintenance, all of it representing resources the body could have deployed differently. The body builds the skull anyway, maintains it throughout a lifetime, and repairs it when damaged. The level of protection afforded to the brain by the skull is comparable to the protection afforded to the heart by the rib cage, and in some respects exceeds it. This protective investment has been maintained across the entire history of the human lineage, and no evolutionary pressure has moved toward reducing it. The same argument extends to the vascular supply. The brain receives blood through a dense and redundant network of vessels, arteries, arterioles, capillaries, fine enough to supply individual clusters of neurons, with backup pathways built in so that if one vessel becomes blocked, others can compensate, and the tissue can sometimes be preserved. The circulatory system serving the brain is more elaborately constructed and more carefully maintained than the supply to almost any other organ. The body treats its continuity as a priority. It has arranged the blood flow and the backup blood flow and the pressure regulation that keeps the supply consistent across changes in posture and exertion, as though the question of maintaining this organ's fuel supply is not negotiable. All of this investment, the vascular elaboration, the protective bone, the disproportionate energy allocation, The evolutionary expansion across millions of years points in the same direction. It points toward an organ that has been at every level of biological organization treated as worth maintaining fully, worth protecting comprehensively, worth feeding continuously. The body's own behavior toward the brain is not the behavior of a system maintaining something 90% dormant. It is the behavior of a system maintaining something it cannot afford to let fail. There is something worth sitting with in the particular texture of that metabolic commitment. The 20% of resting energy that flows to the brain does not represent a lavish excess that evolution simply failed to trim. It represents what it costs to run a system of this complexity at the level of readiness the brain maintains. Every sodium-potassium pump cycling in every neuronal membrane across the whole brain is part of that cost. Every mitochondrion in every synaptic terminal is part of that cost. Every bit of continuous processing happening in the background, maintaining the organized state of the system through sleep and waking and all the hours in between, is part of that cost. The cost is high because the system is large and continuously active and organized in ways that require constant maintenance. Stepping back to the number itself, 20% of resting metabolic energy from an organ representing 2% of body weight, it is useful to consider how specific that number is. Not 15%, not 25%. The figure is consistent across individuals and across physiological conditions that vary considerably in other ways. A person resting at sea level and a person at altitude, a person in a warm room and a person in the cold, a young adult and a middle-aged one. The brain's share of the energy budget is remarkably stable across these variations because the cost of maintaining the system at its baseline is not much affected by most of the environmental changes a person encounters. The demand is set by the organ's internal requirements, not by external conditions. What changes is not the base cost, but the additional draw placed on it by deliberate activity. Reading a sentence calls on different regions than recognizing a face. Reaching for an object calls on different circuits than remembering what happened last Tuesday. Each specific task activates specific regions with measurable precision, increasing local blood flow and local energy consumption in those areas, while the background activity continues across the rest of the brain. The increase is real and detectable. It is also, in relative terms, modest, a layer of task-specific cost added to the constant base. The whole organ is never simultaneously called to maximum effort in all regions at once, but neither is any region ever simply off. The cavefish in its underground river navigates the dark channels with no visual input and no visual processing, its smooth-sealed eye sockets giving nothing away about what happened there across the long generations of its evolution. It moves through complete darkness and seems, by every behavioral measure, entirely adequate to its environment. What its body does not do is maintain structures whose maintenance cost cannot be justified by what they return. The eyes went because they became a cost with no return. The brain of the organism that eventually became human did not go because its cost has been returned continuously, consistently, across every environment and social condition and ecological challenge the lineage encountered. The bill has been paid, every generation, without the system ever running a deficit significant enough to reverse the direction of the investment. The researcher in the anatomy department sets the brain down carefully in its container. She peels off one glove, then the other, and thinks about what she has just handled, not in the abstract vocabulary of neuroscience, though she knows that vocabulary well, but in the simpler terms that the object itself invites. A fifth of every calorie, a lifetime of continuous fuel flowing to this particular piece of tissue, a skull built to protect it. A vascular system elaborated over millions of years to supply it, an evolutionary lineage that expanded it, and expanded it again, and built more bone around the expansion, and kept expanding it, across a span of time so long that the individual generations involved are not imaginable as individual generations, but only as a slow drift in a particular direction. All of that pointed at a structure the size of two fists, sitting quietly in a white container on a lab bench on an ordinary morning, having already, before the day began, consumed its share of the breakfast its owner ate. The cavefish is still in its cave. The rivers beneath the Yucatan still flow through absolute darkness, and the fish still navigate them without sight, their smooth foreheads moving through cold water, their other senses picking up the current and the chemical signals and the pressure changes that tell them everything they need to know. Their bodies stopped paying for eyes when eyes stopped being worth paying for, and now they carry the evidence of that decision in their sealed sockets, generation after generation, in the dark. Evolution is a very patient accountant. It works across timescales that make individual lives invisible, and across the full breadth of that accounting, it has never written off the brain. Just behind the forehead, a few centimeters in from the skin, there is a region of the brain that accounts for roughly a third of the entire cortical surface. It sits closer to the front of the skull than to the back, and in a living person, in the seconds before a difficult decision, its activity increases in a way that can be measured from outside the skull entirely. The region is not performing anything that feels dramatic from the inside. Someone is pausing before they speak. Someone is holding a thought at arm's length and turning it, considering it, putting it down again. In the 2 or 3 seconds of silence before the words come, this particular stretch of tissue, dense and folded and pushed right up against the bone of the forehead, is doing some of the most specifically human work that any organ in the body ever performs. The prefrontal cortex is the name given to that frontal expanse, and it is worth sitting with its size for a moment before anything else is said about it. 30% of the human cortex. That is not a small reservation in the brain's geography. It is more like the whole front country of it, the forward-facing territory where the organ turns toward what is not yet real and begins to work on it. Planning belongs there. The management of competing impulses belongs there. Working memory, the capacity to hold several pieces of information in mind at the same time and manipulate them without writing anything down, belongs there. The restraint that keeps a sharp reply unspoken, the rehearsal of a conversation before it happens, the quiet ranking of possible outcomes, all of it runs through that forward stretch of cortex, processing steadily, largely without announcement. When that region is damaged, the changes that follow are consistent enough to have been documented across more than a century of clinical observation. The most famous account comes from the mid-19th century, from an American railway worker named Phineas Gage, who survived having an iron tamping rod driven through the front of his skull following an accidental explosion. The rod entered below his left cheekbone and exited through the top of his head, passing directly through the prefrontal cortex. He remained conscious. He recovered. His memory, his language, his motor control, all largely intact. What changed was harder to name precisely, but those who knew him described it clearly enough. His judgment was different. His plans dissolved before they became actions. His behavior in social situations no longer tracked the implicit rules that most people follow without thinking. His physician John Harlow noted that the balance between his intellectual capacities and his animal tendencies seemed to have been disturbed. What Gage had lost was not a general mental capacity, but a specific one, and the specificity of that loss pointed directly to where the work had been happening. The prefrontal cortex is not, in any useful sense, dormant in an ordinary person. It activates during the consideration of what to have for dinner when the available options are genuinely equivalent. It activates during the suppression of a startle response. It activates during reading, during conversation, during the management of a calendar, during the half-second pause before agreeing to something that feels slightly wrong. The region that was damaged in Phineas Gage is the same region that flickers to life in anyone who has ever thought twice before speaking or held a plan in mind across the span of an afternoon without writing it down. Moving further back in the brain, sinking a little deeper below the cortical surface, there is a curved structure on each side of the brain, one in each hemisphere, shaped roughly like a seahorse, which is what its name suggests. The hippocampus sits in the medial temporal lobe, and its contribution to the brain's daily work is specific enough that removing it on both sides produced one of the most documented case studies in the history of neuroscience. A man referred to in the literature as H.M., later identified as Henry Malazan, underwent bilateral hippocampal removal in 1953 as a treatment for severe epilepsy. The surgery succeeded in reducing his seizures. It also meant that for the remaining 55 years of his life, he could not form new lasting declarative memories. He could remember things from before the surgery. He could hold a conversation. He could learn certain motor skills through practice without knowing he had ever practiced them before. But the ability to take what was happening now and carry it forward into tomorrow, to move an experience from temporary registration into permanent storage, was gone. What Henry Malazan's case made clear, and what decades of subsequent research has confirmed and extended, is that the hippocampus performs a function so specific that its loss leaves a person intact in almost every other way, while removing the capacity to live in continuous personal time. Every new face he met was, each time, a stranger. Every researcher who worked with him for years was someone he encountered, from his own experience, for the first time. The region was not redundant. It was not doing something the rest of the brain could quietly absorb in its absence. Its work was its own, and when it stopped, that particular kind of work stopped with it. The hippocampus does not work only during waking hours. During the deeper phases of sleep, it generates a characteristic electrical pattern, a sharp, brief surge of activity that researchers record as a wave against the baseline of the sleeping brain. These sharp wave ripples, as they are called, occur in pulses during slow wave sleep, and each pulse corresponds to what appears to be a moment of replay, the neural firing sequences associated with a recently learned experience, repeating themselves in compressed form, the pattern of activity from an event earlier in the day, running again, faster now, in the quieter medium of the sleeping brain. The hippocampus, during these intervals, is not at rest, it is filing. It is moving experiences that arrive during the day toward the longer, quieter corridors of neocortical storage, where they will eventually live without depending on the hippocampus to retrieve them. The work happens without any awareness of it, in a room that from the outside looks entirely still. Deeper in the temporal lobe, beside the hippocampus, sits a small structure that is disproportionately involved in one of the brain's oldest tasks. The amygdala is approximately the size and shape of an almond, and it is present in both hemispheres, one on each side. Its specialization is evaluative rather than declarative. It does not store facts, so much as it registers significance, particularly the significance of stimuli that carry emotional weight. Fear is the response most commonly associated with it, but the amygdala's role is broader than that. It assigns urgency, it signals relevance, it flags stimuli as worthy of attention before the slower, more deliberate parts of the cortex have finished processing what the stimulus actually is. The speed of that response is part of its function. By the time a person consciously registers that something frightening is happening, the amygdala has already sent its signal, and the physiological response has already begun. What happens when both amygdalae are damaged was documented through the study of a woman known in the literature as SM, who carries a rare condition called Urbach-Wieta disease. The condition caused gradual calcification of her amygdalae, and by adulthood, they were no longer functioning. SM's intelligence is intact, her language is intact, her memory for facts, her ability to recognize faces, her social understanding, all largely intact. What she lacks is a fear response. Confronted with scenarios designed to produce fear in nearly any other person, SM reports curiosity rather than alarm. She will handle snakes or approach strangers without hesitation and without the physiological signature of threat response that such situations produce in subjects with intact amygdalae. The deficit is specific. Everything else in her cognition continues as it would in anyone else. What has been removed is the neural structure that in an ordinary person routes certain perceptions toward urgent alarm, and in its absence, that routing simply does not happen. SMDL transbreden to dot happen. Squawkst heppel peculiar kind of clarity. It is not that fear is absent from her world because she is unusually calm, or unusually brave, or unusually experienced with threatening situations. Fear is absent because the structure that generates the fear response is no longer there. The amygdala, in this context, is not an accessory to some other general-purpose emotional capacity. It is the mechanism. Its loss is the loss of the function it was performing, cleanly and specifically, with nothing else filling the gap. At the rear base of the brain, below and behind the cerebral hemispheres, there is a structure that looks from the outside like a smaller brain folded against the larger one, its surface deeply grooved, its texture different from the smooth curvature of the cortex above it. The cerebellum accounts for only about 10% of the brain's total volume. Inside that volume, it contains more than half of all the brain's neurons. The density of packing inside the cerebellum is extraordinary. Hundreds of billions of tightly arranged cells in a relatively compact space, doing something that took researchers a long time to fully characterize. Motor coordination is what the cerebellum was understood to manage first. The smooth continuity of movement, the precise timing that allows a hand to reach for something in the dark and arrive at the right height, the adjustment of posture as weight shifts, the calibration of force that lets a person set down a glass without shattering it. All of this involves cerebellar computation. Damage to the cerebellum produces a condition called ataxia. Movements become jerky, uncoordinated, uncertain. A person with cerebellar damage can intend to perform a smooth action and produce instead something fractured and lurching, the intention intact and the execution disrupted. The timing system has been disturbed, and without it, voluntary movement loses its fluid coherence. More recent research has extended the cerebellum's known contributions beyond the domain of movement, and the extension is larger than initially expected. Functional connectivity studies have established that the cerebellum maintains direct communication with the prefrontal cortex and parietal cortical areas, regions associated not with movement, but with working memory, language processing, and sustained attention. The cerebellum, it turns out, may be doing for thought something like what it does for action, providing the timing and the sequencing, the smooth coordination of successive steps that keeps a line of reasoning on track or a sentence properly assembled. The research is less settled in this domain than in motor control, and the precise nature of the cerebellar contribution to cognition remains an active area of investigation. But the direction of the evidence is clear enough to say that the cerebellum is a participant in more of the brain's work than its position at the back of the skull might suggest, and its density of neurons alone speaks to the scale of computation happening within it. Reaching for a cup in a dark room is a mundane act, and the cerebellum performs its part in it without any deliberate engagement from the person doing the reaching. The arc of the arm is computed. The anticipated weight of the cup is factored into the grip before the fingers have fully closed around it. The arrival of the cup at the lips is timed against the position of the head. None of this is consciously directed. The cerebellum handles it continuously in the background, and then, on the evidence of recent research, possibly goes on to handle something similar for the sequences of thought that follow, the reasoning and the language that unfold in the quiet that comes after the cup has been put down. There is a network in the brain whose discovery came out of an unexpected observation in neuroimaging research. Researchers conducting scanning experiments in which subjects were asked to perform specific cognitive tasks noticed that certain brain regions showed less activity during the tasks than they showed at baseline during the intervals when subjects were lying still, unfocused, asked to simply rest between trials. These regions were not quiet during rest and active during tasks. They were active during rest and relatively quieter during tasks. The pattern was consistent across subjects and across studies. It was not noise. Something was happening during the resting periods that the task periods were, in a sense, interrupting. Acorn Sreichel and his colleagues examined this pattern systematically in the early 2000s and characterized it as a network, a set of regions that activate together in a coordinated way during apparent inactivity. The network includes the medial prefrontal cortex, the posterior cingulate cortex, the angular gyrus, and several other areas distributed across the inner and back surfaces of the brain. Together, they came to be known as the default mode network. Default because these regions represent what the brain defaults to when no externally directed task is being imposed upon it. What the default mode network appears to be doing during its characteristic activation has been reconstructed from careful study of what subjects report thinking about during rest intervals and from the cognitive deficits that appear when the network is disrupted. The network supports autobiographical memory, the retrieval of personal experiences, the sense of a continuous self with a past that can be revisited. It supports perspective thinking, the mental simulation of future events, the rehearsal of scenarios that have not yet happened. It supports social cognition, the modeling of other people's mental states, the attempt to understand what someone else might be feeling or intending, the social reasoning that underlies most of what makes human interaction coherent. And it appears to support a form of creative recombination, the drifting associative thought in which ideas from different domains encounter each other without any deliberate attempt to connect them. When someone lies in a quiet room, apparently doing nothing, and the mind moves without direction from a memory of a summer afternoon to a vague sense of something that needs to be arranged next week to a passing thought about how a friend might be feeling, that is the default mode network at work. It is not idling. The regions that compose it are showing strong coordinated activity. The brain is running its most inward processes, reviewing the personal past, sketching the plausible future, holding the social world in the mind, and turning it in a way that keeps relations with other people legible. The clinical significance of this network adds another layer to its importance. Disruptions to default mode network function have been documented in several conditions that involve characteristic changes in the way a person relates to their own past, future, and social world. In depression, the network shows altered patterns of connectivity, with the ruminative quality of depressive thought appearing to reflect a particular kind of default mode engagement, in which certain content, self-critical, pessimistic, dominates without the modulation that ordinary default mode activity provides. In early Alzheimer's disease, default mode regions, particularly the posterior cingulate and medial prefrontal areas, show some of the earliest metabolic changes visible in scanning, appearing before clinical symptoms become obvious, suggesting that the network's functions are among the first to be affected as the disease begins. In schizophrenia, the relationship between the default mode and task-related networks is altered, with the boundaries between self-referential thought and externally directed cognition appearing less clearly maintained. The network was not doing nothing. It was doing something so consistent and so necessary that its disruption shows up as a change in how a person experiences being themselves. A person lying still in a dim room, eyes closed, apparently resting, has a default mode network lit with activity that a scanning machine would show clearly. The medial prefrontal cortex, the posterior cingulate, the angular gyrus, all brightening together in the characteristic pattern that Reichel and his colleagues identified 2 decades ago. The interior activity is not quieter than it was when the person was solving a problem or reading a sentence. In some respects, it is broader, ranging more widely across time and social space, less focused on a single object, and more engaged in the kind of ranging thought that connects the personal past to the anticipated future, to the felt presence of the people the person knows. It is, in its way, some of the most thoroughly human processing that the brain performs, the part that makes a person, rather than simply a processor of incoming information. The brain's major structures do not operate in isolation from one another. The prefrontal cortex receives input from the amygdala, modulating the urgency signals the amygdala sends, and in ordinary circumstances, shaping the emotional responses that those signals initiate. The hippocampus communicates with the prefrontal cortex during both waking and sleeping, passing along the representations of new experiences that the prefrontal cortex will eventually hold independently. The cerebellum, through its connectivity with frontal and parietal areas, contributes its timing and sequencing functions to the cortical processes those areas support. The default mode network intersects and alternates with the networks engaged during external tasks, the two systems rising and falling in rough complementarity as attention moves between the interior and the exterior of experience. None of these regions is waiting. None is held in reserve for some future eventuality, like an unused room left locked against the possibility of a guest. Each one is engaged, moment to moment, in something that the clinical record has made legible by documenting what happens when it is no longer there. The prefrontal cortex that manages impulse and plan, the hippocampus that holds the bridge between experience and memory, the amygdala that marks the world with urgency and signals what demands attention before the deliberate mind has caught up, the cerebellum that times and sequences and coordinates whether the thing being coordinated is a movement or a thought, the default mode network that keeps the self oriented in its own time and its own social world, drifting from past to future and back again in the intervals when nothing else is being demanded. The clinical neurology that established these functions was built largely from loss. from patients whose injuries or diseases removed the contribution of a specific region and left researchers with the record of what was missing, from Phineas Gage and the railway rod, from Henry Mollison and the epilepsy surgery that traded his capacity for new memory against the reduction of his seizures, from SM and the calcification that took her fear response while leaving the rest of her cognition intact, from the thousands of patients whose strokes, tumors, and lesions mapped the brain's regions onto the deficits their damage produced, each deficit a kind of photographic negative showing the shape of the function by outlining its absence. The logic that runs through all of these cases is the same logic that runs through the architecture of the brain itself. Specific regions doing specific work, work that stops when the region stops, nothing sitting idle, nothing available to absorb the loss without any trace of consequence. The brain described by clinical neurology is not a system with vast untapped reserves. It is a system in which every part is already doing something, and the evidence for that is the long, careful record of what goes wrong when any part of it is taken away. It is worth spending a moment with what that record looks like in its accumulated form. A neurologist's diagram of the brain, annotated with the functions associated with each major region, is in some respects a map of all the things that can go wrong. Damage to the left posterior frontal lobe produces expressive aphasia, the words no longer coming, the grammar no longer assembling, the person understanding what is said to them, but unable to generate speech in return. Damage to the left posterior temporal region produces a different kind of aphasia, the words still flowing, but the meanings slipping, the sentences losing their accuracy, the patient speaking fluently, but in ways that no longer track the world precisely. Damage to the right parietal lobe can produce a condition called hemispatial neglect, in which one side of the perceived world, the left side, is no longer attended to, not because vision in that half of the visual field is absent, but because the attentional system that would normally direct awareness to that side is no longer operating. Patients with hemispatial neglect will eat the food on the right side of their plate and leave the left side untouched. They will draw a clock face with all 12 numbers crowded into the right half of the circle. The left side of the world is still there. It simply no longer registers. Each entry on that map represents a region that was doing something its neighbors could not replicate. The map has no blank spaces. It has no zones labeled inactive or non-essential or available for future development. Every region whose damage is listed in the catalog of neurological deficits is a region that was, until that damage occurred, contributing something irreplaceable to the brain's continuous operation. There is a particular quality to what the prefrontal cortex does that is worth returning to, because it is easy to describe in abstract terms and harder to feel in concrete ones. The region's functions, planning, impulse control, working memory, complex decision-making, sound like formal capacities, the kind of thing that gets evaluated on neuropsychological tests. But the prefrontal cortex is also the region that determines whether a person can maintain a sense of who they are across time, whether they can hold a long-term commitment against the pull of immediate alternatives, whether the future feels like a real place with real consequences, or a vague abstract region that exerts less force than the immediate present. Damage to the prefrontal cortex, in severe cases, does not make a person obviously impaired in the ways that a damaged limb or a damaged eye makes impairment obvious. The person is still present, still responsive, still capable of engaging in the immediate moment. What changes is the relationship between the immediate moment and everything else, the past that provides context and the future that provides direction. Phineas Gage, could still hold a conversation. He could still perform physical tasks. What he could no longer do reliably was let the future constrain the present, let the anticipated consequences of an action shape the action before it was taken. The prefrontal cortex, when it is working, is the organ's forward-looking faculty, the part that holds a longer arc of time in mind and allows that arc to bear on what is happening now. The hippocampus, in turn, is the region that makes the past available to be learned from. Without the capacity to form new declarative memories, a person lives in an eternal present, each moment disconnected from the one before it. The record of experience unable to accumulate into the kind of autobiography that most people carry without thinking about it. Henry Malazan's intelligence remained largely intact. His procedural skills could still be refined through practice, but each day was for him a day without a yesterday. The world continued. The file of his experience of it did not grow. The amygdala's contribution is older in evolutionary terms, operating faster and further below the level of deliberate thought than either the prefrontal cortex or the hippocampus. The signal it sends, this is relevant, this is urgent, this requires a response, arrives before the reasoning structures have assembled a considered assessment of the situation. That speed is part of the design. In conditions where the relevant stimulus is genuinely threatening, the delay involved in deliberate evaluation might be costly. The amygdala roots around the delay by initiating a response before the evaluation is complete, leaving the prefrontal cortex and the rest of the cortex to catch up, and if the threat turns out to be less serious than the initial signal suggested, to modulate the response accordingly. What SM illustrates is not simply that fear is absent when the amygdala is non-functional, but that the system which initiates urgency flagging and rapid response preparation is absent, and that this system does not have a simple backup. The fear response in an ordinary person is not just the subjective feeling of fear. It is a cascade of physiological preparations, elevated heart rate, altered breathing, increased alertness, changes in attentional focus that the amygdala initiates and that the rest of the brain and body then participate in. When the amygdala is gone, the initiating signal is gone. The cascade does not begin. The subjective experience of fear is not produced because the signal that produces it was never sent. The cerebellum, sitting at the base of the brain, with its dense interior of neurons, times these processes as it times the arc of a reaching arm. The research connecting it to cognitive sequencing is newer and less settled than the motor literature, and it carries appropriate uncertainty. But the anatomical connectivity between the cerebellum and the prefrontal and parietal areas is documented, and the functional imaging evidence of cerebellar engagement during working memory and language tasks is present. The picture that emerges is of a structure that was understood for decades, primarily through what its damage did to movement, the lurching, imprecise quality of cerebellar ataxia, and is now being recognized as having additional contributions to the timing and sequencing of cognitive operations as well. The cerebellum is not waiting at the edge of the brain's functional territory. It is participating in it, including in capacities that until recently were not thought to be its domain. The default mode network brings the full picture into a particular kind of relief. Here is a set of brain regions that are most active, not when a specific external task is being performed, but precisely when no such task is ongoing, when the person is at apparent rest, when, by the logic of the 10% idea, the brain might be expected to be at its least engaged. And what those regions are doing during that rest is not nothing. They are maintaining a continuous, metabolically expensive, functionally important process of self-referential thought. the review of the personal past, the simulation of the personal future, the modeling of other minds, the associative drift that connects disparate pieces of experience in ways that have sometimes been linked to creative thought. The brain is not resting when it has nothing external to do. It is turning inward and doing something that requires just as much neural coordination, just as much coordinated activity across distributed regions as any outward-facing cognitive task. If there were a part of the brain that sat idle, if some large territory truly lay dormant through most of a human life, then damage to that territory would produce nothing. There would be no deficit. The person would be exactly who they were before, minus the non-contributing region. But that is not what the clinical record shows. The clinical record shows that damage to virtually any part of the brain produces some specific and consistent change in what the person can do or experience or how they process the world. The changes are sometimes subtle. Some deficits require careful testing to detect. Some occur in domains so specific that a person can live for years without their ordinary life, making the deficit obvious. But the deficit is there, and it points to work that was being done before the damage occurred, work that was quietly, continuously, and specifically happening in that particular region. A building where every room is occupied does not need a reserve of empty rooms to be impressive.
What makes it interesting is not the space available for future use, but the particular activity in each room already underway. The planning taking place in the front rooms, the careful filing happening deeper in, the urgent signaling coming from one small chamber near the center.
The timing and coordination being handled in the dense structure at the back, and the sustained interior activity across a distributed network that in the intervals between external demands draws the self together and holds it briefly in coherent thought. The building is full. It has always been full. Every room is lit from within, doing the specific thing that only that room does, and has been doing it continuously at considerable cost for every hour of the life it has supported. What the brain does when it appears to be doing nothing may be some of the most human work it ever performs, the ranging inward thought that keeps a person oriented in their own life, the quiet review of where they have been, and the loose rehearsal of where they might be going, the half-formed awareness of the other people in the world and what those people might be thinking or feeling. The default mode network brightens during exactly those moments, its coordinated activity spreading across the medial surfaces of both hemispheres in a pattern that is unmistakable in its consistency and its scale. The brain at rest is a brain still at work. The apparent stillness is the surface. Beneath it, always, the same full occupation continues, specific, necessary, and entirely without pause. In the first hour of deep sleep, something begins moving through the brain that has no exact parallel anywhere else in the body. It is not electrical, not the kind of activity measured in spikes and frequencies. It is closer to a tide, a slow, directed current of cerebrospinal fluid pressing through narrow channels that run alongside the brain's blood vessels, carrying with it the accumulated chemical residue of a full day of thought. The channels widen during sleep, the tissue around them having relaxed enough to open a passage roughly 60% broader than the one available during waking hours. And through that wider passage, very slowly, the fluid moves, collecting proteins, clearing debris, carrying toward the brain's drainage systems, the molecular byproducts of everything the organ did while the body was awake and asking things of it. This system has a name given to it in 2013 by a researcher named Micah Nettergaard and her colleagues, who described it in the journal Science after years of watching it function in mouse brain tissue. They called it the glymphatic system, borrowing the prefix from the glial cells that form the walls of those perivascular channels, the cells whose swelling and contraction help regulate how freely the fluid can move. The name was new. The process was not. The brain had been running this quiet nightly maintenance long before anyone had the tools to see it happening, long before anyone understood why a night of poor sleep could leave a person feeling not just tired, but dimly wrong, in a way that was hard to name, that particular cognitive fog that settles not in any one faculty, but everywhere at once, a general loss of sharpness whose source was, for a long time, genuinely unclear. What the glymphatic system removes is not trivial. Among the proteins it clears are amyloid beta and tau, molecules that, when they accumulate in the spaces between brain cells over years and decades, appear in the tissue of people who develop Alzheimer's disease. A single night of disrupted sleep measurably increases the concentration of amyloid beta in the cerebrospinal fluid. The glymphatic current moving through its channels in the dark while the body lies still is doing something that matters across a lifetime. Not dramatically, not urgently, the way a harbor cleans itself between tides, steadily, without announcement, as part of the ordinary rhythm of use and restoration that keeps the whole system functional. The image that stays with it is simple, an interior dark and quiet, and through it a slow current moving. No fanfare, no particular drama in the motion itself, just fluid pressing through tissue in the hours after midnight, carrying things away, making room, leaving the architecture of the brain slightly cleaner than it found it. This is one of the things the brain does during the hours that to a person looking at someone asleep appear to be the hours when nothing is happening at all. Sleep, when examined closely enough, turns out to be among the most structured things the brain ever does. It does not simply stop. It reorganizes. It moves through stages with a consistency that, once mapped, revealed something researchers had not fully anticipated, that the different stages are not merely different depths of unconsciousness, but different operating modes, each serving functions that the waking brain either cannot perform or performs only partially. Slow-wave sleep, the deep phase that dominates the early part of the night, is when the glymphatic current runs most freely. It is also when something else is happening in a structure tucked into the medial temporal lobe that has to do not with cleaning, but with keeping. The hippocampus, during slow-wave sleep, produces what electroencephalographers call sharp-wave ripples, brief, intense bursts of coordinated neural activity lasting only a fraction of a second, originating in the hippocampus and spreading outward. They occur in a particular rhythmic relationship with two other signals, slow oscillations that roll across the cortex in long, gentle waves, and shorter bursts called sleep spindles generated by the thalamus. The three signals interlock, arriving in a specific sequence, repeating across the hours of slow-wave sleep with a regularity that suggests not accident, but coordination, something organized, something functional, something the sleeping brain is clearly doing on purpose, even though purpose is a word that requires some care when applied to an unconscious organ running a program that predates human self-awareness by a very long time. What those ripples contain, or what they appear to contain, is memory. Not memory in the abstract sense, not the concept of remembering, but the specific firing sequences that represent particular experiences encoded during the preceding day. A route walked, a face seen, a word learned in an unfamiliar language. The hippocampus holds these recordings temporarily, the way a field recorder holds audio before it is transferred to a more permanent medium. During slow-wave sleep, those recordings appear to be transferred, gradually across many nights, with each night's ripples contributing to the slow redistribution of newly learned information from the hippocampus into the broader cortical networks where long-term memories are eventually stored. The process has a name, though the name is less important than the image. Memories moving in the dark, the organ redistributing its holdings, while the person who owns those memories lies still and does not know it is happening. The evidence for this process comes partly from studies in which slow-wave sleep was selectively disrupted, not by waking subjects fully, but by playing quiet auditory tones precisely timed to interrupt the slow oscillations without crossing the threshold into waking. Subjects who underwent this disruption performed measurably worse on declarative memory tests the following day than subjects whose slow-wave sleep was left intact. The disruption was targeted, so was the deficit. The specificity of the relationship between the disrupted process and the specific kind of memory that suffered afterward is the kind of experimental result that makes a phenomenon feel not just observed but understood. There is something in that finding that quietly dismantles a particular assumption, the assumption that the sleeping brain is simply the waking brain, with the lights off, idling in a low-power state, doing nothing of consequence until morning. The assumption is intuitive. Sleep looks like nothing from the outside. The body is horizontal, the eyes are closed, the breathing is slow, and the person is by the ordinary definition absent. But the ordinary definition, it turns out, describes the outside of something whose inside is conducting some of its most careful and consequential work. Later in the night, the character of the brain's activity shifts. The slow oscillations of deep sleep give way to something faster, more varied, more closely resembling the brain's waking state. The eyes begin moving beneath their closed lids. The body remains still, actively held still, in fact by a mechanism in the brainstem that suppresses motor signals to the voluntary muscles, preventing the sleeper from acting out what the brain is experiencing. And the brain, during this stage, is experiencing quite a lot. REM sleep takes its name from those rapid eye movements, first recorded and described by researchers in the early 1950s. What the EEG showed during REM was, to the researchers who first saw it, genuinely unexpected, not the slow synchronized waves of deep sleep, but a high-frequency, low-amplitude signal almost indistinguishable from the trace produced by an alert waking brain. The brain was, by every electrical measure, running at full capacity. The person was asleep. The body was still. The brain was not resting. What activates during REM is a particular constellation of regions, the visual cortex, brightening with images assembled from memory and imagination, the amygdala, running at levels comparable to or exceeding what it produces during emotionally significant waking moments, the anterior cingulate cortex, engaged with the kind of activity it performs when evaluating complex or charged situations, the prefrontal cortex, the region most associated with rational oversight, and the evaluation of what is and is not real, shows relatively reduced activity compared to its waking state. This particular pattern, emotional and sensory regions running bright, executive regions running quiet, is thought to be part of why dreams feel so thoroughly convincing while they are happening. The machinery that would ordinarily flag an impossible sequence of events as impossible is, during REM, operating below its usual threshold. The dreaming brain accepts what it generates. It has no particular reason not to. Dreams have been documented across every human culture with a recorded history, and in most of them, they were treated as signals from somewhere beyond the dreamer, messages, warnings, revelations. The neuroscience does not entirely dismiss the instinct behind that interpretation. even while relocating its source. Something real is happening in REM sleep. The brain is not producing noise. It is processing emotional experience, consolidating procedurally learned skills, and running something that looks very much in its patterns of activation, like a simulation, a rehearsal space where events can be experienced and their emotional weight can be felt without any of the actual consequences. The interpretation of what that is for remains genuinely open. The observation that it is happening, and happening with specificity and structure, is not a different kind of evidence for what sleep accomplishes, comes from a study involving a rat, a maze, and a set of electrodes placed in the animal's hippocampus. In 1994, Matthew Wilson and Bruce McNaughton at the University of Arizona published recordings that showed something that had not been clearly documented before. As the rat ran through the maze during the day, specific hippocampal neurons fired in specific sequences, each neuron corresponding roughly to the rat's location as it moved, so that the animal's route through the maze was legible in the pattern of its own brain activity. Then, later, as the rat slept, the same neurons fired again, in the same sequence, in the same order, at roughly the same relative timing, only compressed, the whole route replayed in a fraction of the time it originally took to run. The sleeping brain was rerunning the maze, not randomly, with the specific pattern of the specific maze the specific animal had run that specific afternoon. The phenomenon is called neural replay, and it has since been documented in many species and across many kinds of learning, not just spatial navigation, but motor sequences, perceptual patterns, procedural skills. In studies where replay during sleep has been measured and correlated with subsequent performance, the relationship is consistent. More replay, better next day performance on the tasks whose neural patterns were replayed. The sleeping brain, running these sequences in the dark, appears to be practicing consolidating the motor and cognitive representations that will allow the skill to be performed more fluently when the animal or person wakes and tries again. There is something gently interesting in what that implies. A musician who goes to sleep after a difficult practice session, frustrated by a passage that would not come together, may wake to find that the passage sits more easily under the fingers. The improvement happened during the hours of apparent inactivity. The skill was being worked on while the musician lay still and dreamed of something else entirely. The brain did not stop when the practice session ended. It continued the work in a different register, using a different method, in a different state, and it did this whether or not anyone thought to ask it to. The specific costs of disrupting this nighttime work were mapped in careful detail by Hans van Dongen and David Dinges, whose research tracked subjects sleeping either 6 hours or 8 hours per night across 2 weeks. The results from the 6-hour group accumulated quietly. Each day, performance on cognitive tests declined slightly, reaction times slowed, working memory errors increased. The cumulative deficit, by the end of 2 weeks, was equivalent to what a person shows after staying awake for 2 full consecutive days. The striking detail in the study was not just the magnitude of the impairment, but the subject's own assessment of it. Their subjective sense of sleepiness plateaued and stabilized after a few days, even as their objective performance continued to worsen. They felt, at the 2-week mark, not very much worse than they had at day 4 or 5. But their test scores told a different story, one they were not, at that point, in a position to fully read, because the very faculties that would have allowed them to notice the impairment were themselves among the things that had been compromised. The brain's account of its own state, when that state includes significant sleep debt, is not fully reliable. This is not a criticism of any individual person. It is a feature of how the system works, and it applies broadly. The organ doing the evaluating is the organ that has been compromised. The measurement tool and the thing being measured are the same instrument, and the instrument has drifted without registering that it has drifted. What remains legible to someone looking at the data from outside is that the nighttime work the brain performs is not supplementary. It is not a bonus available to people who choose to prioritize it. It is part of the operational cycle of the organ as necessary to its daytime function, as the daytime activity itself is necessary to making the nighttime work worth doing. This is the picture that emerges when the brain's sleeping hours are examined with the same care that has been applied to its waking ones. Not an organ at rest, an organ that has changed what it is doing, from processing and responding to clearing and consolidating, from directing attention outward to directing it inward, from making decisions to reviewing and reinforcing the patterns that make decision-making possible every night, in every sleeping person, in a sequence as structured as anything the waking brain produces, and as necessary as any of it. There is something that the brainstem does during sleep that deserves a moment of attention, not because it is dramatic, but because it is so completely continuous. The brainstem maintains respiration, it manages heart rate, it regulates body temperature, adjusting minute by minute in response to signals from the periphery and the environment. These functions do not pause during sleep, and they do not hand themselves off to the sleeper's conscious oversight, because the sleeper has no conscious oversight to offer. They run on their own, as they have run on their own since long before the cortex existed in anything like its current form. Older structures performing older functions with an unbroken reliability that is, when considered quietly, one of the more remarkable features of being alive. The brainstem during deep sleep is as active as it needs to be, and what it needs to be is quite active, and it does this without any of the credit that tends to go to the more photogenic regions of the brain, the ones that light up in colorful images during language tasks and moral dilemmas and the recognition of faces. During sleep, the cerebellum also continues its postural work, making small adjustments to the sleeping body's muscle tone, participating in the regulation of movement during REM sleep in coordination with the brainstem's motor suppression system. The visual cortex processes the images that constitute dreaming. The thalamus, that deep relay station, coordinates the traffic of signals between cortical regions and subcortical structures across every stage of the night. The anterior cingulate remains engaged. The amygdala pulses with something that, in functional terms, resembles the processing of emotional significance, even in the absence of any real event to process. The whole organ is occupied. None of it is dormant. The lights are on in every room, though the rooms are being used for different purposes than they serve during the day. The 10% figure, if applied to the sleeping brain, would be even harder to defend than when applied to the waking one. There is no hour of the night in which 90% of the brain goes quiet. There is no stage of sleep in which the majority of the organ powers down and waits. The glymphatic system is running its slow current through the tissue. The hippocampus is firing its sharp wave ripples in sequence. The brainstem is holding the breath steady. The visual cortex is generating scenes. The amygdala is processing them. The cerebellum is managing the body's position in the bed. The thalamus is coordinating signals across the whole distributed system. 10% is not a number that describes any of this. It is not a number that describes anything the brain does, in any state, at any hour of the day or night. What the brain is, when all of this is considered together, is not well described by percentage at all. It is better described by what it actually does, which is to say, it is an organ that is always doing something, even when that something is not visible, not legible to introspection, not accessible to the person carrying it around. It is running cleanup operations whose importance accumulates across decades. It is rehearsing motor sequences whose consolidation shows up as improved performance on the next day's attempt. It is redistributing memories from a temporary holding structure to the wider networks that will carry them for years. It is maintaining the body's respiratory rhythm with a consistency that has not faltered in a lifetime of use. It is doing all of this simultaneously in a space that weighs roughly 3 kilograms in the dark without waiting to be instructed. The myth of the dormant brain persisted as long as it did, partly because the tools for seeing inside the living brain were slow to arrive, and partly because the myth told a story people found useful. There was something encouraging in the idea that most of the brain was waiting, that untapped capacity was not just possible, but guaranteed, sitting in reserve like an inheritance not yet claimed. The story was generous. It made the listener feel that they were, by definition, more capable than they had yet managed to demonstrate, and that the gap between their current performance and some ideal performance was not a matter of effort or limitation, but of access. All that unused brain waiting to be switched on, but the brain that the instruments revealed when the instruments finally arrived was nothing like that. It was not a bank of dormant circuits waiting for a key. It was something more like a city that runs continuously, that has districts performing different work at different hours, that maintains itself at considerable metabolic cost, not because it can afford to be profligate, but because the cost of maintaining is lower than the cost of shutting down and restarting, and because every part of the city is doing something that the rest of the city depends on, even the parts whose function is not obvious to someone driving through on a single afternoon. The sleeping city is still a city. The lights in certain districts dim while others brighten. The overnight infrastructure runs: sanitation, power distribution, the slow work of maintenance crews moving through streets that are, during the day, too crowded to service. The people who live there are not conscious of most of this. They sleep through the part of the city's daily operation that is, in some ways, the most necessary part. They wake to a city that is, because of what happened while they were not watching, ready to be used again. This is approximately what happens inside the skull between midnight and dawn in a person who is sleeping, the glymphatic tide moving through expanded channels, the hippocampus pulsing its small bright replays into the waiting cortex, the amygdala processing the emotional residue of the day just past, the brainstem holding the breath, and the heartbeat with its ancient, reliable constancy. All of it happening at once. All of it happening in the dark. All of it happening, whether or not the person whose brain it is has any awareness that it is occurring. The brain does not wait for acknowledgement to do its work. It does not require the person to understand the glymphatic system, or to know the name of the hippocampal sharp wave ripple, or to have any sense at all of what slow wave sleep is accomplishing in the hours before the dream stage begins. It runs its programs in the absence of any observer. It has been running them since long before anyone had language to describe them, or instruments to measure them, or journals in which to publish the measurements. It ran them in the brains of every human ancestor who ever slept, in every cold or warm place, across every climate and every era, without deviation and without rest from the rest. And it will run them tonight in the dark as the body grows still, and the breath slows, and the particular kind of attention that holds a person alert to the world begins, gradually to release its grip. The sharp wave ripples will begin their small coordinated pulses. The glymphatic channels will widen and admit their slow tide. The visual cortex will in a few hours begin assembling its particular version of elsewhere. The brainstem will hold the breath as it has held it every night, with a patience that has no name because it does not need one. The brain at 3 in the morning, in a quiet room, in a sleeping person, its glymphatic current moving in the dark, its hippocampus holding and slowly releasing its day's recordings. Its brainstem steady as something that has never once forgotten what it was doing, is not an organ at reduced capacity. It is an organ fully occupied with the work that this particular hour requires. Every region doing what this hour asks of it, every system running, the whole interior lit in its own way, going quietly about its business, as it has every night in every sleeping person across the full long span of time that human beings have been sleeping at all. And somewhere in that span, someone first suggested that most of it was unused, that a large portion of this continuously humming, metabolically expensive, intricately organized organ simply sat idle, waiting for the right stimulus or the right course of self-improvement to activate it. The idea traveled far. It appeared in books and advertisements and motivational speeches and films. It gave people something to reach for. The brain, meanwhile, kept running, every region cycling through its functions, every neuron maintaining its membrane potential, every synapse awaiting its signal, the glymphatic tide moving in the dark long before anyone had given it a name or understood why it mattered. All of it continuous, all of it in its quiet way, rather a lot. The slow oscillations of slow-wave sleep roll across the cortex like something unhurried and very old. They have a rhythm. They do not rush. In the hours when the glymphatic current is moving most freely, when the hippocampus is most actively distributing its temporary holdings into the broader cortical networks, and the brain is also at its most thoroughly resting, in the sense that the sleeping body has withdrawn from the demands of the world, released the tension in its muscles, surrendered its alertness to stimuli, and given the organ that manages all of that its best opportunity to do the maintenance work that waking hours crowd out. The rest and the work are not in conflict. They are the same thing, observed from different angles. The brain rests by working differently. It recovers by doing the things that only the dark and the stillness allow it to do. This is not a paradox, exactly. It is more like the difference between what a river does during a flood and what it does between floods. Both are the river being a river. Both involve water moving through a landscape according to its nature. One is more visible. One accomplishes things that the visible phase makes necessary. The quiet phase is not less real for being less dramatic. The brain at the end of the night, having run its glymphatic clearance, having fired its hippocampal replays, having moved through its cycles of slow wave and REM sleep with the regularity that characterizes a healthy sleeping brain, is not the same brain that fell asleep. It is slightly cleaner in the chemical sense. It is better organized in the representational sense. The memories encoded during the previous day have moved a little further along the process of consolidation. The motor skills practiced the day before have been rehearsed internally in ways that will make them more fluent when they are attempted again. The emotional residue of the day's significant events has been processed by the amygdala and the anterior cingulate in ways that are not fully understood, but that appear, on the evidence, to matter. People's emotional responses to difficult events are typically less acute the morning after than they were the night before, and there is reason to believe the brain's overnight processing is part of why. The brain wakes into a world that is, because of what it did during the night, slightly more ready to meet, not completely ready, not perfectly organized, but better than it was when it closed the day. The overnight work was real work. Its results are real results. They are just not the kind of results that are easy to see from the outside, which is why for so long the hours of sleep were understood as hours of nothing, and why the brain that occupied them was thought to be an organ at reduced engagement, doing little of consequence until the eyes opened again. The instruments that revealed otherwise did not change what the brain was doing, they only made it visible. The glymphatic tide was moving before Mike and Nedergaard's laboratory had the tools to see it. The hippocampal sharp wave ripple was firing before Matthew Wilson and Bruce McNaughton placed their electrodes in the sleeping rat's tissue and recorded the maze being quietly replayed. The REM brain was running at its full waking frequency before the first EEG trace showed researchers a signal they initially assumed must be an artifact, because surely a sleeping brain did not look like that. All of it was always happening. The seeing came after. The doing was always there, in the dark, in the quiet, in the hours when nothing appeared to be happening at all. There is something gently settling in that. The brain's work does not depend on being witnessed. It does not require a scanner or an electrode or a published paper to proceed. It runs because it runs, because the organism that depends on it has always depended on it running, and because the structures that run it are very old, and because the processes they execute have been refined across a span of time so long that the whole of human recorded history is a small fraction of it. The brain sleeping tonight in any quiet room is running a program that was already ancient when the first human being first noted that sleep seemed to matter without knowing why. The cerebrospinal fluid moves through its channels. The hippocampus sends its small ripples outward. The brainstem holds the breath. The dream begins its careful, unregulated, emotionally real assembly somewhere in the visual cortex and the amygdala, and the dreamer does not know that they are dreaming because the part of the brain that would know is running, tonight, at a lower threshold than usual, and the tide moves on, and the replays continue, and the breath continues,